Hi everyone, my name is Kelvin Pogo, and today I'll be talking about transcription. So, transcription is the synthesis of RNA to DNA. Sorry, the synthesis of RNA with the information of DNA, right? So, yeah, let's go. Let's look at it. So, there's three stages involved in transcription: initiation, elongation, and termination, right? I think they look pretty much easy for my level. All right, so this is how the whole initiation part begins. So there's a region on the DNA helix, right, called a promoter, right? And when it comes to eukaryotes, there's one promoter that is very crucial called the TATA box, all right? So yeah, so on the promoter, with the help of transcription factors, all right, RNA polymerase will bind to it, then the initiation of transcription begins, right? So yeah, so let's say if this is the helix, right? This is the region, the promoter region, right? RNA polymerase. I need some transcription factors here. I need certain amounts, not only amount. I need transcription factors to invite me to do my binding, right? So I have my transcription factors here. RNA polymerase, I bind to it. Then initiation of transcription and that's the beginning of initiation, right? So after initiation, we move to the next stage, which is elongation, right? So elongation, this what happens in elongation. So this is the double helix, right? Double helix. I think this, yeah, it mirrors. So this is the promoter region, right? I bind to it, then this is what the RNA polymerase does. It untwists the double helix, right? As it untwists, also moves, right? So as it's moving, it makes nucleotides, right? Nu- Not just nucleotides. It picks this strand, right? This strand, which is the template strand, right? Then it makes the complementary nucleotides, right? So which, yeah, I think this gives a better picture of it. So yeah, the RNA polymerase untwists the double helix, then it makes um the nucleotides, right? And nucleotides are added to the 3 prime end of the growing RNA molecule, right? So this is how it's going to look like if it was in codons, right? So it'll look pretty much like this. I need a test. So 5 prime TT C slash G A C slash that's then GGG. All right. Then don't forget. Then there's the 3 prime. Never forget. All right. So this is what um the RNA polymerase would do. It make a complementary base. So yeah, that means this is going to be 3 prime and TT, right? So what is complementary to T, which is A, right? So A, that's then complementary to GC. Then A, we are talking about RNA. So in this part, it wouldn't be T, right? It will be USL, which is U. So CUG, right? Then CCC, then the 5 prime, right? So that's what happens in the elongation part, right? So it keeps making it, and yeah, the RNA polymerase it keeps moving, right? It keeps moving on the DNA double helix, right? It keeps moving on it, keeps moving on it. So that's what it is for the elongation part of transcription. Oh, and don't forget this part. Yeah, this is the mRNA, which is being made from the template, right? The template, which is shown here. Yeah, this is the template. Yeah, the template of the DNA. So, all right. So in the termination part, which is the last part of it, right? What happens is remember when I was talking about the promoter region, right? There's another region called the terminator. So the RNA polymerase gets to the terminator, and what happens is so yeah, let's use this again, right? Right? So it's already on twisting, right? It's moving, moving, moving. Oh, sorry, moving. Then it gets to the terminator. So remember, as it moves, right, it's on twist, then it twists it like it twists the thing back again, right? So RNA polymerase is responsible, basically. He fixes his own mess up. Yeah. So when he gets to the terminator part, right, what happens is it deassociates, right? And when it does that too, the mRNA also moves, right? It also disassociates from the RNA polymerase, right? So all these things happens in the nucleus, right? Then the mRNA, yeah, like I said, depends on the type of cell you're talking about. But yeah, so in the end, termination, there's the disassociation. Sorry, there's the this association of RNA polymerase and the RNA. So that's it for the termination. So that's pretty much everything. Yeah, which yeah, you could actually see it from here. So yeah, this part goes and that's pretty much it. Thank you.